Good afternoon to all participants. Welcome to the PharmTalk 1, the webinar series. This event is conducted monthly by School of Pharmacy Institute Teknologi Bandung, so don't forget to follow our social media account for the updated information. Today's webinar topic is new receptors, new bioassays, new ligands, medicinal chemistry of Orphan G protein coupled receptors, which will be presented by Professor Dr. Renat Krista M. Müller from Department of Pharmaceutical and Medicinal Chemistry, Pharmaceutical Institute, the University of Bonn, Germany. Today's session will be led by Dr. Renat Rahmana Emrad Kartasasmita from Department of Pharmacochemistry, School of Pharmacy, Institute Teknologi Bandung, Indonesia. Dear participants, before we start, allow me to deliver the ground rules for today's webinar. Your microphone will be automatically muted. You will have the opportunity to engage with the speakers and moderator and each other via an online chat room. Do not post any requests for technical support in the chat room. Chat rooms can only be used to give topic-related questions to the panelists. Do not provide your feedback about the quality of the session in the chat room. You will be given the opportunity to do this by completing a survey after the webinar conclusion. You are encouraged to use the virtual background designed for this webinar. And in the middle of the webinar, the attendance list will be given in the chat room. So please carefully fill your name in the form, as your field name will be attached in the certificate. To begin this agenda, we are going to hear a welcoming speech from the Dean of School of Pharmacy, Institute Teknologi Bandung, represented by the Vice Dean of Academic Affairs. We would like to invite Dr. El Fahmi to deliver this speech.
Okay, thank you very much, Mrs. MC. Okay, good afternoon, everyone, and good morning for Professor Krista Müller in Germany. So first of all, on behalf of our Dean, the Dean of School of Pharmacy, I would like to welcome you, all of you, in the webinars and the School of Pharmacy organized the webinar series, and this is the first one, what we call PharmTalks 1. So of course, we hope this meeting will be available for us. So actually, we would like to have the offline meeting, but due to the pandemic COVID is still there, so we are not able to organize the offline meeting. So we would like to optimize our seminar, with organize this seminar, the webinar, and hopefully that all of participants can learn from Professor Krista Müller. And we know that Professor Krista Müller have very interesting research in medicinal chemistry, especially for the receptors, ligand and receptors, and we can learn a lot about this lecture from Professor Krista Müller. And then this lecture, of course, will be chaired by our colleague from School of Pharmacy, Dr. Emrad Kartasasmita. And for our information, that's our juniors, Dr. Andika, right? Andika is working in the Professor Krista Müller. So this is also very good initiation for good collaboration. I think that can be further developed in the next future. So without any ado, I would like to thanks all of you for this seminar, and of course, thank you very much for Professor Krista Müller. We really appreciate for your sharing in this very, I think, very fruitful online meetings. So I think this is all what I would like to say for the opening speech in this moment. Thank you very much for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. El Fahmi, for the welcoming speech. And dear participants, without any further ado, please welcome Dr. Renat Rahmana Emran Kartasasmita.
Thank you very much, Ms. MC. Before we continue with the presentation of Professor Müller, first of all, I would like also to say very nice good morning to you, Professor Müller in Germany, and also good afternoon to all audience in Indonesia. So before we start with the presentation, I would like also to read the CV of Professor Müller. So Professor Müller completed her PhD studies at the University of Tübingen in Germany in pharmaceutical and medicinal chemistry in 1988. And after that, she follows a postdoctoral stay at the NIC in United States. And after her return to Tübingen, she completed her habilitation and became associate professor of pharmaceutical chemistry at the University of Würzburg in Germany in 1994. And since 1998, she holds a full professorship at the University of Bonn in Germany. From 2001 until 2004, and from 2010 until 2015, she served as vice rector of the University of Bonn. Currently, Professor Müller is the vice president for research of the German Pharmaceutical Society since 2019, and president of the German Urine Club since 2015. She served also as associate editor for RCC Medicinal Chemistry and as a member of several scientific and editorial boards of other scientific journals. She has published more than 450 scientific papers, including patents. Among several other prizes she received, the first is the Nauta Award for Medicinal Chemistry and Chemical Biology by the European Federation of Medicinal Chemistry in 2018. And in 2019, she received the Tony Holy Lecture Award in Prague, the Czech Republic. Her main interests are focused on medicinal chemistry, of course, and pharmacology of purine-binding membrane proteins, that is receptors and enzyme, orphan GPCRs, and G proteins. That is the short CV of Professor Müller. And so shortly we will hear the very interesting presentation of Professor Müller. So Professor Müller, the time is yours, and you have around 50 minutes for the presentation, and we will continue with the discussion session. Please, Professor Müller.
Thank you very much. Dear Dean, dear professors, dear colleagues, dear students, researchers in Indonesia, welcome to my talk. Selamat datang di presentasi saya. Unfortunately, my Indonesian is not so good, so I will speak in English. And I will speak today, as was already announced, about orphan G protein-coupled receptors and about new receptors, new bioassays, and new ligands that we are developing. I'm very proud actually for the invitation, and I'm also very, very happy, I must say, to have Andika Maradika here in my group. He's a brilliant PhD student, and he's working in the field of orphan G protein-coupled receptors. So let me start to introduce to you G protein-coupled receptors. I think everybody knows them, but just a short reminding of their properties. They are 7 transmembrane domain receptors. They have these 7 transdomain helices, which span the cellular membrane, and they are very important for signal transduction, for chemical signal transduction. So a chemical signal will arrive, and then the conformation of the receptor will change, and then a signal is transduced into the cell. And GPCRs, G protein coupled receptors, they are the major drug targets. More than 30%, some people even estimate that even more than 40% of the marketed drugs, and also many drugs that are currently in development, interact with G protein coupled receptors. Interestingly, for many of these GPCRs, we have 800 G protein coupled receptors, more than 800 in the human genome. And for many of them, no ligand and no drug has been developed. So this is an area which is underexplored. So there's a lot of opportunities to develop drugs for G protein coupled receptors. And more than 100 of these GPCRs are orphan, which means that they do not have a ligand. We do not know their ligand, their physiological ligand. So we were thinking, and many people think that the development of drugs targeting these orphan receptors is of very, very high interest. And here you can just see a slide which you probably know. GPCRs, why are they so interesting? Because they can be targeted by many different types of ligands, by small molecules, even by calcium, for example. There's a GPCR which is activated by calcium, by lipids, by peptides, proteins, and so on. And if they are activated, they activate this heterotrimeric G protein, which consists of 3 subunits, alpha and beta gamma. Beta gamma is always together, and activation causes a dissociation of alpha from the beta gamma, and alpha has some effects, and there's different subunits. Beta gamma has different effects within the cell, and this is very important. So if we look at the phylogenetic tree of the G protein coupled receptors, we have more than 800 sequences, as I already mentioned. We have different classes. So the biggest class are the rhodopsin-like receptors. This is class A, and they are shown here. Class A are the blue ones. Here, this big part. So this is further subdivided into alpha, beta, gamma, and delta branches, as you can see here, different branches. And we are mostly interested in the delta branch, which is shown here, the delta branch. And if you see these flags, these are the X-ray structures that have been determined. And this is not completely up to date, but as you can see here, the delta branch is poorly investigated. So this caused our interest, because we want to investigate something which is new, which has not been investigated already by many other people. So there's a lot of things to discover in this delta branch, and that's why we focus on this delta branch. This delta branch receptors, they are activated by a large variety of endogenous ligands, lipids, peptides, nucleotides, and most importantly, they are involved in inflammation, immunity, and cancer. And this is a field where we are really interested in finding drugs for treating cancer, for activating or inhibiting the immune system, and treating inflammatory conditions. And our current main interests are in some of these orphan receptors. I will talk today about GPR35, G protein coupled receptor number 35, and GPR84. Andika, for example, is working on GPR18, which is one of the most amazing orphan G protein coupled receptors. Some are numbered like this, and some have other names like MRG, PRX, mass-related G protein-coupled receptor X, and this is another field we are working on. I want to tell you a little bit more about these orphan G protein-coupled receptors. So orphans are receptors whose endogenous, whose cognate or physiological ligands in the body are still unknown. And the nomenclature, as I just mentioned, is mainly by numbering, GPR1 and so on, or some of them have different names like trace amino associated receptor or mass-related G protein-coupled receptors. And after the physiological ligand, this is just a picture to show this to you. What I'm meaning, this is the ligand and this is the receptor, symbol for the receptor. After the ligand has been discovered, and we are also trying to discover the physiological ligands of orphan GPCRs. So we try to de-orphanize these receptors. After the ligand has been discovered, the receptors are called adopted receptors. They are no longer orphans. But a very important problem is that this de-orphanization is often wrong. So you see this pairing fits, but if you look at this pairing, something is wrong, right? So very often people, scientists, they write publications, they claim to have found the physiological ligand for an orphan receptor, but later on it turns out to be completely wrong because maybe the concentration is not high enough of this ligand that has been proposed, or the affinity is very low, for example. So many of the so-called de-orphanized GPCRs, orphan GPCRs, are not really de-orphanized because the pairing has been wrong. Okay, what are we doing? What is our main interest? We are medicinal chemists, and we are interested in developing tool compounds. So we develop compounds that are high quality tools, and we name them if they are useful, and we recommend them for use to other labs, biologists, pharmacologists. We give them a number, PSB number, Pharmaceutical Sciences Bonn, and a number. So you will find in the literature, if you look up in the internet, you will find a lot of PSB compounds which have been developed over the years in our lab and which are used by other groups for pharmacological studies, for research. But we are also interested in the targets, and these are in this case, the receptors. So we are interested in their structures, in the binding site, in interactions between the compounds and the targets, and in the activation of the receptors, which is associated with the conformational change, or in the inhibition of the receptor. We develop label tools, which means radioactive tool compounds for radioligand binding assays, or fluorescent labeled tool compounds. And we are also interested in studying the pathophysiological roles of the receptors and to perform target validation studies. Is a certain receptor suitable for targeting by drugs? And more during my career, I've been interested in doing drug research, which means developing drugs in collaboration with industry in the advanced phases. And we more and more also file patents for our compounds that we have developed. Okay, what are our methods? We are medicinal chemists, and as medicinal chemists, you have to do interdisciplinary research. You have to have a lot of different techniques in place. So what we do is we express our target of interest, our receptor, in cell lines. And we usually express them in mammalian cell lines like CHO, Chinese hamster ovary cells, or HEK, hamster embryonic kidney cells, or in astrocytoma cells, the human cell line, or in insect cells if we want to purify them and crystallize them later on. We usually use retro and lentiviral expression systems because they give a high expression level in these mammalian cells. We select monoclones because if you have polyclonal cell lines, you lose the receptor after some time, after some passages. We perform mutagenesis studies and prepare chimeric receptors in order to identify the binding site of our ligands. So this is the biology side. Of course, we have a lot of compounds, we synthesize compounds, we collect compounds. And since many years, since I did my PhD, actually, I started collecting compounds. And at one point, I had a lot of boxes with compounds. I did not find anything anymore. So I decided to establish a proprietary compound library. It's our Pharmaceutical Sciences Bonn compound library. And I will show you, I will get back later on to the previous slide again. This is our compound library. It is really professionally managed. And we have a barcode system, only limited access, only by some people, because otherwise the compounds will disappear with time. We have a computer system, a software, which is organizing the compounds, and we link in the database physicochemical and biological data of these compounds. It is compatible with industrial platforms, which is very important for collaboration with industry. So if we want to collaborate with industry, we have to exchange data and we have to exchange compounds. And this is very easy with our system. Of course, we have freezers, minus 80-degree freezers for some compounds, and we store also solid compounds, different batch sizes, also DMSO stock solution in these freezers. So, and our compound library has already been audited by our collaboration partners from industry. So it's really professional, and we can find every compound, everything, and also the annotated data. This is really important for a group like ours, which has a big collection of about 30,000 compounds. And we have natural products. We also collect also extracts, mixtures of compounds, and of course a lot of synthetic compounds. Then we have the assays. So we develop assays like radioligand binding assays, fluorescent assays, nanobread assays, modern assays. These are binding assays, but also functional assays, like G protein couple dependent assays, cyclic AMP assays, for example. Or now we have modern G protein activation assays, which are based on fluorescence or luminescence. And we also perform a lot of beta-arrestin recruitment assays, which I will explain later on a bit more. We do our type of high throughput screening, which means sometimes we screen several thousand compounds at one time. And of course, if we find or develop potent compounds, selective compounds, we do a very broad characterization of these selected compounds. And finally, we do molecular modeling, receptor modeling, pharmacophore modeling. And recently we have started to do X-ray structure determination of G protein coupled receptor. This is not easy, but we have already obtained the first X-ray structures now in our group. And if you have a good structure, you can also do virtual screening, which is can be really, really successful. So these are our methods. And of course, in the center of our work is medicinal chemistry, is the synthesis and optimization of compounds, which I will present you also today for 2 orphan receptors, as I mentioned before. Okay, this is just a selection of tool compounds. I told you that the good compounds that are useful are labeled PSB, and previously, before I came to Bonn, I was at the University of Würzburg, and before that in Tübingen, we had different names for these compounds. But this is just a selection of compounds which are used by the scientific community. And for example, recently I looked up PSB 603 in the internet. You can see that many companies, at least 6 to 8 companies, are selling this compound. This is an A to B adenosine receptor antagonist, and this is broadly used. This is one of the current standard compounds. We did not patent it, unfortunately, at that time, but this is good, it's useful, and the scientific community is using actually this compound. And as you can see here, we have fluorescent labeled compounds, and we also have radioactive labeled compounds developed and used by people. Okay, let me start with the first G protein coupled receptor, GPR84. It belongs to the rhodopsin superfamily, class A, and the subbranch in this case is unclear. It has some relationship with adenosine and biogenic amine receptors, and it's activated by fatty acids, medium chain fatty acids, and hydroxylated fatty acids. But it's unclear whether these are really the physiological agonists, because you need very high concentrations in a micromolar range to activate GPR84. It couples primarily to the GI protein pathway, and what caught our interest, it is expressed mainly in the immune system, in the bone marrow, in leukocytes, in monocytes, and the expression is actually upregulated upon infection. LPS, lipopolysaccharide, this is a constituent of bacteria cell wall, and if you have a bacterial infection, GPR84 will be upregulated. You have a much higher amount of this number of these receptors in this case in monocytes, immune cells, also in the brain, in immune cells of the brain, in microglia. And this is very interesting because it looks like this receptor has a pathological role. So it has a role in disease. And that's why we started working on this receptor. So agonists could be of interest for stimulating the immune system, for a defense against infection, maybe against coronavirus infection, who knows, or for the immunotherapy of cancer. And this is a hot field right now, that you activate the immune system in order to kill the cancer cells, which is already very successful with some compounds, and now other principles are being identified and searched for, and this could be one receptor which could be interesting in this interest in this context. Also, antagonists could be interesting just for the opposite. If there is too much inflammation in the body, neuroinflammation like Alzheimer's disease, multiple sclerosis, and so on, or some other inflammatory diseases, antagonists could be of interest. And so this is the receptor again, GPR84. So it activates GI proteins and reduces cyclic AMP concentrations in the cells. On the other hand, if you have here strong stimulation for extended times, the receptor is phosphorylated here. It recruits beta-arrestin, and this causes an internalization of the receptor. The receptor is no longer in the cell membrane, and this stops the signal by an agonist. So this is the termination of the signal. And this can also be used for testing agonists. Okay, so when we started our study on GPR84 agonists some years ago, there was, in the literature, there were these 3 classes of compounds: fatty acids, very weakly potent, then these fatty acid-like compounds, but without a negative charge. These are deprotonated at physiological pH value of 7.4, so they are negatively charged. And this is a uracil derivatives with a long alkyl chain. It's neutral, but it mimics these fatty acids. And there was a second class, diindolylmethane derivatives. And DIM itself is a natural product I will show you in a minute. And this diindolylmethane was shown to be a pretty potent agonist. And this shows cyclic AMP accumulation, phosphocholine stimulation of the receptor, and then inhibition by the agonist. And you can see here we get a nice concentration response curve, but only in cells which are transfected with the human GPR84. So you can see here this is the non-transfected receptor which doesn't get any signal. And you can see here also that decanoic acid, which is was proposed as the physiological agonist, is much weaker, only a very weak signal. So we worked on this scaffold, and I just show you that this is a natural product. Actually, it is formed from glucobrasicin, this compound, which is present in vegetables. These are vegetables which are very common in Germany. We eat them a lot, especially now in winter. We have winter time. Today is not so cold, but 2 days ago it was snowing heavily. And so these winter vegetables are eaten a lot. They are very healthy and they protect from cancer. They are known to protect from cancer. And this is due to these compounds which are formed from glucobrasicin, which is a glucoside. Okay, so it is transformed to indole 3-carbinol, and in the stomach, gastric acid, it is dimerized to diindolylmethane, 10 to 20%. This is the main metabolite, and there are some other compounds that are being formed in the stomach. So this compound is a potent GPR84 agonist, and we decided to do a program to synthesize analogs, derivatives. So we modified all the different positions. We introduced different substituents here, and also on the indole residues, we replaced carbon atoms by nitrogen atoms here, oxidized the compounds in different positions, and obtained a lot of different diindolylmethane derivatives. And just to show you a little bit of synthesis, we optimized synthetic procedures. For example, if you want to get symmetrically substituted compounds, you can just start from the substituted indole and treat it with formic acid in water at 100 degrees in the microwave, and you get a high yield of the symmetrical compound. We optimized this procedure and also introduced substituents in the linker carbon atom here. And you see the optimization, we could do it in 5 minutes at room temperature in water with very, very high yields. So this is a much optimized procedure, which is a green chemistry actually, just in water. Okay, but if you are a medicinal chemist, you know that the binding sites are never or rarely symmetrical. So we wanted to have unsymmetrical DIM derivatives, and so we had to develop a new reaction procedures to have different residues here and here, and we did it starting from this activated compound, ammonium derivative. This can react in the 3 position. This attacks nucleophilically here at this carbon atom. And this reaction was optimized regarding solvent, temperature, and so on for one specific derivative. And then we synthesized a whole series of compounds. And you can see that the best conditions were water, again, green chemistry, water at 100 degrees centigrade, and no acid, no additive, no catalyst is necessary. We have a very broad substrate scope and high yields, and we got unsymmetrically substituted dim derivatives. Now I show you the results of the biological assays. So I told you that we do both G protein coupled signaling, we investigate, and beta-arrestin recruitment. And some compounds, some agonists actually have a specific property. They show biased signaling. Biased signaling means they do not activate both pathways equally, but they have a preference. And if you want to have an agonistic activity, you want to have biased compounds towards G protein signaling. So they will never recruit beta-arrestin. They will not stop the signal. So you have continuous signaling, okay? Because you, in this case, you want to activate the receptor. You do not want to stop the signal. And actually, this tetrafluoro diindolyl methane derivative was found to be a very potent biased compound. It is much more potent in the nanomolar range, EC50 value 41.3 nanomolar for activating G alpha I, while it was much less potent in inducing beta-arrestin recruitment. So this is a biased agonist. We also found in the same series compounds which do not have a large bias. They are, this compound in the center is similar in both pathways. And we also found a compound which prefers here beta-arrestin recruitment. This means this is a functional antagonist. It stops the signal immediately, and this will block the receptor. Okay, we then went on to investigate a second scaffold, 6-octyl aminouracil, and we did a lot of variations. I only show you a very small part of what we did in this case. Namely, we optimized it. We found that a hexyl residue is better. This is a very potent compound, PSB 1584, very potent agonist. And we made a radioligand. So we synthesized a compound which has a double bond here. And then we did catalytic hydrogenation with tritium gas to obtain tritium labeled radioligand. And this can now be used for radioligand studies, radioligand binding studies. So we can do association studies to the receptor. We can perform dissociation studies and investigate the kinetics. And we can do competition binding. So this is just the cold compound, non-labeled compound, which competes for the radioligand. You get a nice concentration inhibition curves using cell membrane preparations which express GPR84. And we can show that this radioligand really has a very high affinity, and it shows reversible binding. So you can do competition studies, low nanomolar range. So a very good tool to do binding directly to the receptor. And we did this with a series of compounds. I just show you that we have a lot of compounds that compete, that show this nice competition curves. For example, this dodecanoic acid here, weakly active, but it competes with the radioligand. So it means that it binds to the same binding site, this one, as our radioligand. This compound, for example, was very potent here, shown here, and some other compounds. And then we looked at the diindolyl methanes, which we had synthesized and which I had shown before. You can see here that they show a completely different pattern. They increase even the binding of our radioligand. So they are allosteric modulators. They are positive allosteric modulators. They activate the receptors by themselves, which means they are agonists, but they also increase the binding of these lipid-like agonists to the receptor. So they clearly bind to a different binding site, which was really interesting to see, and which was possible to show by by preparing this radioligand. Okay, and we also did some modeling then finally, and this is the allosteric binding site that we propose. Just at the entrance of the receptor, and the lipid-like compounds bind here. Right now, we do not know where the D-indolyl methane derivatives are binding, but they certainly do not bind in the same site, probably at some other place where they have an effect on the conformation of this pocket and increased binding of the allosteric agonists as well. So what I showed here in this field, we successfully optimized agonists from 2 different classes of compounds, lipid-like and di-indolyl methane. And meanwhile, we also developed antagonists, which I'm not showing today because of the lack of time, but we have also competitive antagonists which bind to this binding site, block the receptor, do not activate it, but prevent the activation by lipid-like agonists. Let me now move on to my second receptor, GPR35. GPR35 is a delta branch G protein coupled receptor. Very interesting. It is there are 2 isotypes which have the same pharmacology. We did not find any difference. And there are some endogenous agonists postulated, like quinolinic acid shown here, but we know that this is extremely weakly potent. So it is definitely not the right agonist. And we believe that this receptor is still orphan. So another ligand has been postulated, like this CXCL17 or this lysophosphatidic acid, but we could not confirm the activation of the receptor by these lipids. So we do not think that they are the physiological agonist. So this receptor is also involved in immune reaction, in modulation of the immune system, pain sensation, and in wound healing. And when we started our project on GPR35, there was no potent or selective agonists available. And this receptor is also GI protein coupled and also coupled to G13, similar to GPR84. And I want to show you now our beta-arrestin assay, because this assay is really useful for screening, because false positive results are very rare. It is very specific for the receptor that you are studying, because you modify the receptor, you attach to the receptor a protein which is part of a beta-galactosidase. So this is an enzyme complementation assay. You have this enzyme which doesn't exist in human cells, in mammalian cells, beta-galactosidase. Part of this beta-galactosidase is attached to the receptor recombinantly, and part is attached to beta-arrestin, okay? And when beta-arrestin is recruited to the receptor, the beta-galactosidase becomes active enzyme, and it can then, if you add this substrate, it can cleave the substrate, and this causes chemiluminescence, and you can measure the luminescence, you can measure light. So if the receptor is activated, it forms this enzyme and you get a signal. You can also look for inhibition of the receptor by activating it first with an agonist and then blocking it by an antagonist. Okay, so what we did a screening campaign. We tested compounds from our in-house compound library, which I showed you before. We did screening of a lot of compounds, and we got 3 hits, essentially. You can show the curves here. We did some concentration activation curves. And we had different scaffolds. And actually, we selected the weakest one. This blue curve here, we selected this compound because it's very small. You can see it's a fragment. And also because we were thinking that we could easily modify this structure. The other ones, they have carboxylates, which are not very good for if you have two carboxylates, maybe too acidic, too polar. And this compound, we had some analogs and we could not find any structure-activity relationships. Also, this small fragment has the highest activation of the receptor. So we systematically modified this compound in different position. For example, here, this position, we found out that oxygen is very good. Here's no oxygen, no activity. I must say that this is the p-EC50 value from the beta-arrestin recruitment assay, which means the higher the bar, the more potent the compound. Okay, so we increased the potency, and the best one was a benzamide residue in this position. So we continued with this benzamide. We introduced some residues in different positions. The best one was the para position. And you can see here that the methoxy group was best. So we get already low micromolar potency or submicromolar potency. We get already in the nanomolar concentration range, p-EC50 of about 6.5. So we continued with this position. So this is the 6th position, and we introduced different substituents, keeping this best substituent here in the 8th position. Okay, so small residues were tolerated for especially electronegative lipophilic residues, and bromine was really good. So we now already have a nanomolar agonist here. So halogenation was best. And you can see here the concentration response curves of these agonists. This was our starting point, and you get a left shift. This is the second compound in the low micromolar range, then with a methoxy group already in the sub-micromolar range. And then we have a compound which is really potent, 12 nanomolar. And this was the best compound from this series. We then tested for selectivity. We investigated at the most closely related receptor, GPR55 is very closely related. And we found that it is extremely selective. So it's 3 orders of magnitude selectivity, higher potency here at GPR35 than at the human GPR55. So the next step was to make a radioligand here, this compound, which we achieved by introducing a tritiated methyl group by methylation of the phenolic OH group. And we could do some saturation binding assay. It works like a charm, very good radioligand, KD value of 5 nanomolar. And then we, this is actually the first radioligand for GPR35 that has ever been developed. And it is still the only one. And then we looked for different species. This is a very important point. If you want to develop tool compounds, you need to make sure that they are also active in mouse and rat, because usually pharmacologists, they use mouse or rat models for testing the compounds. You cannot immediately start to work in humans to do clinical trials. And in this case, this was really disappointing because our compounds shown here, they were much weaker in rat and almost inactive in mouse. And this is a big problem, so you cannot use them for mouse studies. And we tried to develop then compounds which are also active in mouse. We switched to a new scaffold which was shown to be a little bit better in mouse, different substituent in this 8 position. And we optimized this compound, this scaffold, systematically by introducing different substituents again in the 6th position. Bromine was pretty good already. Cyanation in this position improved the potency further. And we then increased here the residue. And we found out that if you introduce alkyl chains, you get a further improvement in potency. This is still at the human receptor, and then we checked for rat and mouse. You can see here we get already better, at least at the rat. We already have pretty good potency, but the mouse is still not satisfactory. So we had to think to improve it further on. We increased further on the chain length, and we also did some, we introduced a 3rd ring. So this fixes the conformation. It gets less flexible, this residue, by obtaining this ring. It's then rigid, and you can fix the bioactive conformation. Actually, this was very beneficial. Again, we found that alkyl residues in the 6th position, which corresponds to the 6th position of our chrominones, are very well tolerated. And then we increased the chain length. And we see that with increasing chain length, we get extremely potent compounds. And now, please check for the data at mouse and rat. Now we have the first compound, which is super potent in the low nanomolar range, and it's equipotent in all 3 orthologs. It's equipotent at human, rat, and mouse. And this can now be used for mouse studies with a Ki value of 6 nanomolar, so extremely potent agonist, even for the mouse GPR35. Okay, so this is the summary of what we found out at the GPR35. We identified a new lead structure, we optimized it, we developed the first radioligand, and we developed the most potent agonists that are known so far. And they are even potent at the rat and at the mouse orthologs and can be used for animal studies. So this brings me to the conclusion. I hope I could show you that G protein coupled receptors are fascinating and important protein targets. We have a large variety of receptors and ligands, and we can discover innovative drugs for these GPCRs. X-ray structures and cryo-electron microscopy have a large impact nowadays. Did not show that, but they are very important for virtual screening and for drug design. And GPCRs have a highly complex signaling. There's biased signaling, as I showed you. We have crosstalk, we have receptor heteromers. I did not talk about that today, but it's more complex as you would think. And the more we know, the more complex the picture becomes. Well, I think that often GPCRs are a really promising area for future discoveries, especially in the field of immunology and cancer. And well, they are really attractive new drug targets. De-orphanization is not easy, and very often the proposed pairing is not correct. So fatty acids are probably not the real agonist, the physiological agonist for GPR84. But we could develop new tool compounds for GPR84 and also for GPR35. And these allowed the development also of new assays, which then again result in novel compounds. And this allows target validation studies, which is the prerequisite for drug development for these targets. Let me finally thank my group. So we are a big group, and these are the people who did all the work in this area that I presented today. We have some important collaborations, and of course, funding is very important. I would particularly like to point out that Andika Maradika, who is from your university, we are really very happy with him. He's excellent, brilliant scientist, and I hope we can collaborate later on when he's back at your university in Indonesia. With this, I thank you very much for your kind attention. Terima kasih. Thank you.
Thank you, Professor Müller, for a very interesting presentation, and you finish your presentation also on time. This is typical Germany, very on time. Now, I think we will continue with the discussion session. So if anybody from the audience has any question, you can raise your hand from the, I think there is feature in the Jones application where you can raise your hand. So I still didn't see any question, but I see there is also a question in the chat. Maybe I will read one of the question in the chat room. This is from the Agustina, Agustina Nur Sahyanti. I don't know from which institution, but the question is, how about GPCR, GPRC 5A and GPRC 68? Both of them may contribute to the malignant phenotype. Are you also working on these two GPRs? Thank you. That's the question.
Thank you very much, Agustina, for your question. These are really interesting GPCRs. Right now, we are not yet working on this. Maybe in the future. Well, there is always a history why we start on a certain receptor class. And, you know, my history is I did a postdoctoral stay at the National Institute of Health in Bethesda, Maryland, in the USA. And there I started working on purinergic signaling and purinergic receptors, adenosine receptors. And now we sorry, there's some noise. And now we started first working with compounds, with receptors that are related to purinergic receptors, P2Y receptors, or related to adenine receptors. So we started from that, but now we are expanding. And for example, GPR18, on which Andika is working intensively, we started because our colleague was working on cannabinoid receptors, and he asked us whether we can do something on cannabinoid receptors, and GPR18 is a cannabinoid-like receptor. So there's always a history, and so far we not get in touch with your receptors that you are interested in, but maybe in the future, especially GPR68 I think is on our list.
Okay, I think the next question is from Francisca. Oh, but I lose your question, Francisca. Can you ask directly to Professor Muller? Francisca, I cannot read your question in the chat room.
I can read it.
Yes.
This is a brilliant question. Thank you, Francisca. You asked, diindolylmethane is derived from a natural product, and it is an agonist at GPR84. And you asked, does it have activity at cannabinoid receptors? Since the indole moiety is known as a core structure for cannabinoid receptors. And sure, yes, I can tell you that Andika is actually working on characterizing all of our diindolylmethane derivatives as agonists or antagonists at cannabinoid receptors. And DIM itself, the unsubstituted one, is actually more selective for CB2. It's an agonist, partial agonist of CB2, but we also have different activities, and we are just writing a publication. We just submitted a publication, the first publication, where we showed some effects of diindolylmethanes on cannabinoid receptors. Yes.
Okay, thank you, Professor Muller. The next question is from Siti Para Rahmawati. Please, you can direct the question. You can ask the question. Okay. You are still stumped. Muted, I mean.
Yeah, thank you. So thank you very much, Professor, for your very informative, interesting presentation. So I am a pharmacologist and interested in characterization of ligands. So in your experience, is there any way to characterize a ligand function as an agonist or antagonist other than or before performing functional studies such as pro or anti-inflammatory proliferation and so on? And I heard that you mentioned briefly about allosteric or orthosteric binding, if I'm not mistaken. And yeah, since I'm not familiar with this, can you maybe elaborate more about this? Thank you.
Yes, thank you very much for this very good question, which gives me the opportunity to talk a little bit more about biology. Because we can only do in our lab in vitro biology studies. This is one of our main shortcomings, I would say. So if we want to do animal studies, we rely on collaborations because we do not have animals in our institutes. So we are very much interested. So we do everything that we can in vitro, and we characterize agonists, allosteric agonists or orthosteric agonists by different types of assays. So we have really a big range of in vitro assays, like, for example, cyclic AMP. So we measure the second messengers or calcium measurements. And we also do mutagenesis studies to try to find out whether it's to which side it is binding on the receptor. And also we do some co-crystallization studies recently. But we cannot do in vivo studies. And this is something which is, we are very much interested in collaboration. So in vitro, we have good possibilities to do these studies. As far as it is limited to recombinant cells. If it goes to primary cells or, for example, immune cells, we also cannot do it very well in our lab. We are not experts in this. And for this, it would also be interesting to collaborate. So we have a lot of different assays, as I mentioned, besides binding studies. Sometimes you can also do, with radioligand binding studies, you can also see whether it's an agonist or an antagonist by adding GTP, for example, to the binding assay, or sodium to the binding assay, because you get a shift for agonists sometimes in these assays. I don't know whether I answered your question, but very interesting. And if you are interested in collaboration, we are very open to collaboration.
Thank you, Professor Muller. There is a next question in the chat room from Apertis, from Miss Apertis or Mr. Apertis, I'm not sure. How about orphan GPCR may contribute to immunology, or its mechanism? Or is its mechanism maybe?
Ah, okay.
Yeah,
Okay, this is a good question, actually. How does it work? How do they affect immune cells? So first of all, they are highly expressed. Some of them are highly expressed on immune cells. And the signaling is complex, but the signaling actually Particularly may work via cyclic AMP. It may also work via kinases, certain kinases. In the end, the effect that you can measure is that you have cytokine release, for example, IL-2 release or interferon gamma release or something like that. But it's a complicated system. May I also answer a question from the chat, which was about fragment-based approach? Enade Istia Stono asked, what do you think about fragment-based approach to work in the GPCR de-orphanization? Thank you for this brilliant question. I think this is a good idea. I think fragment-based approaches in general, also for drug development, are really useful, are very good, because you can grow the fragments. And also for de-orphanization, it makes a lot of sense to start screening small fragments, find an effect, and then look for derivatives, analogs. And yes, definitely. There is one little problem, maybe. Sometimes these receptors are very specific and exclusive. This means that if you change a small thing like a hydroxy group, it gets completely inactive. Okay, that's the main problem if you use this fragment-based approach for de-organization. That very often you really need to have the final compound in order to see an effect. Otherwise, you don't see anything. It's not true for all receptors, but for some. GPR 183, for example, is such a receptor. You need really the full steroid to have a strong effect, to have a real effect. Thank you.
Okay. Yeah, thank you, Professor Muller. Thank you for the question, Mr. Enade. Anybody has new question? Maybe direct question to Professor Muller or via chat? So, Mr. Daryono, please, Mr. Daryono, you raise your hand. Mr. Daryono, please, you can ask your question.
Okay, thank you very much. First, I would like to thanks to Professor Muller to give a very nice lecture this afternoon, morning in Germany. Yeah, from your lecture, you told that the response of your design molecules to the GPR35 in humans, rats, and mouse is different. That's why you then optimize or makes the orthologs to fit or to get best structure for most GPR35, for example. My question is there any significant structure of the GPR35 humans, rats, and mouse? Thank you.
Yeah, actually, well, there's no X-ray structure, and you have to do modeling, which is not so easy for GPR35, which is there's no related X-ray structure from a related receptor available, so it's rather difficult to actually identify the binding site. So we have done some modeling. We've tried to do it, and you see large differences between the three species. You know, several amino acids are exchanged, which explains. But I did not show any model because it's very hypothetical. You know, we have no evidence for the structure. But the differences in sequence can easily explain. Sometimes it's only one amino acid which makes a huge difference, you know, for the binding. And this happens very often with receptors. And, you know, nowadays everybody is screening for the human target. And this is a big problem. And then they use it's published, people use the human ligand to study it in mouse, and they are surprised that they don't see an effect. But it's not possible because the compounds are inactive. Yeah.
Okay, thank you.
Okay,
Important
thank you.
problem.
So, anybody else who will ask a question?
Maybe I can share again the
Oh, yes, please.
screen just to let me see. Okay, I have one slide which I want to share with you because I'm an associate editor for RSC Medicinal Chemistry. This is from the Royal Society of Chemistry, a journal on medicinal chemistry. Oh, sorry, I don't know what happened. Sorry, something. Oh, no, sorry. This is okay. Sorry, and I want to invite you to, if you have a nice paper, original research, or review article. We welcome nice papers, also by young scientists, by everybody, to submit to our journal. So it has a new name. Previously it was MedChemCom, and since last year it's called RSC Medicinal Chemistry, and we accept small papers, full papers. So everything, you are very welcome to submit to our journal.
Okay, thank you for the information, Professor Muller. I see now somebody who raised hand. Miss Damayanti. Please, Miss
Thank you,
Damayanti.
Evan. Hello, Professor Muller. I don't know if you still remember me. I was by Professor Kasak in 2005 until 2007, and then we moved to Düsseldorf. So it's nice to see you again. I was in your classes many times, and still inspiring as before for your lectures. So my question is actually a bit practical. So we have Andika there in Bonn, and hopefully he can finish soon. So what do you think? What he can develop in Indonesia with his research in Bonn? Because maybe we have a constraint with the instrument and so on, or maybe you can suggest how he can start in Indonesia, continuing the topic that you already developed in Bonn. Thank you, Prof. Muller.
Thank you. I will discuss with Andika. I have not thought about it so far deeply, because what Andika is doing, part of it is quite expensive and difficult to do, but I'm sure there are some things he can do there which is easy for him to do there, also with limited instrumentation. I do not know your lab, so I guess you have also pretty good conditions for working. And you know what I have been always interested in, and this is something which is on my list. I visited Indonesia when I was very young, student.
You're still young now.
And I know a bit of Indonesia, Java and Bali. And I love the country. It's a beautiful country. And I know these jamus. And I have always been interested in investigating these jamus, their constituents. And I think that's a nice thing to do, to work on the pharmacology of these Indonesian bioactive drug preparations that you have in Indonesia. And you can do some pharmacology on them, vitro pharmacology. And I think Andika has great potential. He can learn new techniques, he can develop further. He's almost done with his. We are very happy with him, as I mentioned before. And I think he can do good things there. We can also discuss it here and think about something. I cannot give you a full answer now because I have not thought about it, and also because I do not know your lab.
Yeah. You should visit again Indonesia then. Okay, thank you, Prof. Muller.
And see you in Bonn again.
Yeah, I hope so. I was there in 2015, but I went to Düsseldorf to meet Professor Kasak. No chance to Bonn. Okay. Yeah, bye-bye. Thank you, Pak Emran.
Okay, thank you, Ms. Damayanti. I think there's a question also from Tasya. Yeah? It is a general question. Could you please elaborate more about how to design compound library, Professor Muller?
How to design what?
Could you please elaborate more about how to design compound library?
Compound library. Okay, yes. I think a compound library is really important. I recommend you to do the same. Actually, I tell this in many talks about our compound library, and I have got a very good feedback from, for example, in a Polish city, they did it because I gave a talk 10 years ago. They told me later on they did a compound library, collecting from all your colleagues, because it's a treasure. Compounds are a treasure, and when I did a PhD, my colleagues, when they finished their PhD, they threw away their compounds, they discarded them, and it hurt me. I could not do it with my compounds. I still have my compounds for my PhD, and so please collect it, and I can give you advice to do it specifically. It's not too expensive. You can start on a simple level. I started already almost 20 years ago. And we started slowly, and then we got a funding, and we could do it in a better way. We could hire an informatician. We did a collaboration with a colleague from informatics and so on. And there's a lot of software, freeware available that you can use and adapt. So just start collecting, and you can visit. I visited companies, big companies, small companies, before I started establishing the compound library.
Okay, thank you, Professor Muller. So there is another question from Ms. Safitri. Thank you for very interesting talk. Many GPCRs have been found to dimerize or have interacting partners that change receptor signaling. Do you have any idea if orphan receptors may also do the same?
I'm sure that they do the same. I'm pretty sure that they do the same. And we actually we have had some master theses looking into that as well, but it's a huge field. Where do you start? So we start with the homomeric receptors, and later on we move on. If we have a suspicion, we look at heteromers. I'm sure they do. I'm sure they do, and it is another level of modification. It's very important. It's the future thing to investigate all this network.
Okay, thank you, Professor Muller. One more question from Ms. Amelia, Tasya Amelia. How to differentiate bias signaling to bias due to error or misinterpretation in assay?
Yeah.
A little bit technical question.
Thank you. I see you have a lot of experience in this field already. Yes, this happens very often, and you have to do a strict control. So you also need to have compounds which are non-biased. You have to observe the whole range of biased, non-biased, and so on. And there are some controls. You need to have controls, and there is you have to calculate it, complex way of calculation. Whether it's biased, what is the bias factor or not. So control essential.
Okay, thank you. So I think from the time we still have around 10 minutes left for discussion. So if still any question from the audience, please. So I didn't see any question from the chat room, and also nobody has raised the hand. Maybe a short question from me, Professor Muller. From a lot of legions that we have synthesized, is there any compound that has finished the preclinical study, so in animal, complete until preclinical, so that have the potential also for clinical study.
Yes, thank you for this good question. Yes, indeed, we had collaboration with the company, and we finished all the preclinical studies, and now the company is looking for, it's a medium-sized company, and they're looking for a big partner to do the clinical studies. So it is, we did several patent family with the company, and they did a lot of animal studies with our compounds. We have synthesized more than 1,000 compounds, and we have not published, only patents so far. We are waiting for the next step, and it depends, it always depends on different factors, because clinical trials are extremely expensive, and so they are looking for a strong partner. It also depends always on different factors, like how convincing are the animal studies? How is it's also the economic situation? Is this a preference or not? They have, let's say, they have 10 candidates which can go to clinical trials. They only have money and capacity for 2, so they do prioritization. They postpone it a little bit. So we are waiting for what most promising one.
Okay, thank you. Anybody else? We still have a question. We still have around 6 minutes time. So from the audience, okay. Anybody? Please raise the hand. Oh, still one. Yeah. This is a question in the chat room. Sorry.
Yes. Yes, after internalization, you asked.
Yes.
Iris, Jenny, thank you. I would like to know what happened to PR84 after internalization through arrestin signaling by the antagonist, by the agonist. It's actually an agonist. Was it degraded through autosomal degradation? Yes. Okay. So the receptors have different abilities. They are internalized, but some of the receptors, they can go back to after a cycle, so it's a small cycle, they can go back to the membrane after a short period of time. They are internalized by activation, and then they can move back to the membrane after maybe 20, 30 minutes. And some are degraded by proteasomal degradation. But the body, the cells, they are usually economical. Okay? So if possible, they will save the receptor, and it gets sometimes also inserted into membranes of internal organelles. And are probably also signal internally. This is an area, there are some publications that GPCRs can continue signaling when they are internalized, because they are in the membrane of organelles like mitochondria or other organelles. Okay, so this is still pretty much not, well, it's not a complete black box, but still a lot to research, and each receptor is different, behaving differently. But there is some economy that not everything is degraded. Only a small percentage is degraded because it's a lot of energy which the cell needs for degradation and for resynthesis. And the organism tries to avoid that if possible.
Still have around 3 minutes time. Maybe a question from somebody from audience. So in chat room, no question, also nobody who raised the hand. Maybe a last question from me, Professor Muller. Yeah, maybe not directly related to the presentation today, but I used to see your presentation in YouTube about the super caffeine, which has the potency as the anti-dementia, anti-Alzheimer. What about this compound? Is there any further development from this super caffeine? Because I drink coffee every day, maybe three times.
Yes, I started with adenosine receptors. Caffeine is acting via adenosine receptors. It blocks A2A. It blocks all adenosine receptor subtypes, and A2A, the A2A adenosine receptor, is particularly important for many effects, positive effects of caffeine. And I did it already. I started already in USA to work on this, and this field has developed. As you probably know, one compound has been approved for Parkinson's disease now, cadephyllin, which is also an A2A selective antagonist, and it stops, not stops, but it decreases the chronic development of Parkinson's disease, of neurodegenerative diseases, probably also of Alzheimer's, at least in animal studies. So our compound has unfortunately not been patented. That's our problem. Okay, we are working on other ones still. We are still working on A2A antagonists. We collaborate with a group in China who do the animal studies. And we will see. Now also A2A antagonists are in clinical trials, as you know, for cancer, because it activates the immune system, it fights cancer. And so actually we can recommend to drink coffee and also tea. I know that in Indonesia people drink also a lot of tea. So teh botol, I know.
Yes.
and both is good. I mean, it contains caffeine. Only for children and for pregnant people or nursing mothers, they should not drink caffeine or very little only. But otherwise, it is positive, and it's completely been well. Previously, it was recommended not to drink it, but now pharmacologists have a completely different view because there are clinical studies, huge epidemiological studies, which are positive for that. So keep drinking three days.
Yeah, thank you. Yeah, in any cases, I need a cafe every day.
Yeah, I drink tea, caffeine in the morning and tea during the day.
Okay, thank you for your suggestion. And also our time is up. We are now exactly 4:30, yeah, and I think we have to finish this afternoon session in Indonesia and your morning session in Germany. Thank you very much, Professor Muller, for very nice presentation and interesting discussion. So, ich danke Ihnen für die interessante Präsentation and Diskussion. Ich spreche noch ein bisschen Deutsch, Professor Muller, auch nach 20 Jahren.
Sehr gut, sehr gut.
Yeah. So please give applause for Professor Muller, for our audience from the park. Thank you very much. And I return the session to the MC. Please, Ms. Aziza. Thank you.
Okay. Thank you so much for Professor Dr. Renat Krista M. Muller for the wonderful presentation, and also Dr. Renat Rahmana Emran Kartasasmita for leading the discussion. So dear participants, this webinar is about to come to an end. So before we end this meeting, we would like to invite all participants for group photo session. So please activate your video and use the virtual background given via email. Okay.
Beda. Iya, nggak kelihatan.
Okay, I will count to 3.
Tapi backgroundnya beda ya, nggak bisa ya.
Okay, I will count to 3, and Okay, 1, 2, 3. Okay, how about the committee? Is it okay? Okay, next slide. So please smile again. 1, 2, 3. Okay, please wait a minute. Okay, and next slide. So it's about 7 slides, yeah? So please be patient to hold your smile. Okay, 1, 2, 3. Okay, thank you. Next slide. 1, 2, 3. Okay, next slide. 1, 2, 3. Okay, so is that all? The comedy? Have you got? Okay, so I think that's all. Oh, one more. Okay, hold your smile. Smile to your camera. 1, 2, 3. Okay, thank you so much for all the participants of this Farm Talk. Thank you so much for your attendance. So see you on the next Farm Talk. This event is conducted monthly by School of Pharmacy Institute of Technology Bandung. So don't forget to follow our social media account for the updated information. So see you on the next event. Thank you so much for all the participants. Okay, don't forget to fill your attendance form, yeah?